Good morning or good afternoon, or actually good evening, like we have today here in Wolfenbüttel. Okay, welcome back to to Coffee Break with TRANSFAC. And today we will have quite interesting topic to discuss. And as usual, let me share the screen. As usual, you can ask questions anytime. Just type them in this Q&A section here in Zoom, and I will try to interrupt and answer a question if you have questions to the talk or to the presentation. Actually, my plan today is to give you a very quick recap of what we discussed before, and with a bit more focus on the topic today, and then I will go online to demonstrate all this what I have done here on the slides to you, how we can do it in our platform or using our tools, and then we have enough time to answer all your questions. And yeah, then you are, as usual, welcome to ask as many questions as you like. Okay, we'll start from this slide, and as you can see, the topic today is TRANSFAC pathways. What is it? I mean, TRANSFAC, you know, it's a database to predict binding sites, but it's not the end. So as you can see here, the questions we're asking is, okay, fine, you found your sites in your promoter, so what's next? What is next? Actually, we have other tools in our portfolio, in our platform. We have a database called TRANSPATH. We have other databases like publicly available, like GenBH, Reactome, HumanSeq, and we integrate this with our tools, and which means, and you'll see how after you found your sites and you found your transcription factors, which are important, you can go further to talk about, to see more about pathways. And actually, we will also do a little detour in Cytoscape to see how those pathways can be visualized also in the other tools. And okay, let's go ahead. I'm sorry, I got a little bit cold. That's why I'm drinking my favorite tea now, not coffee. So we have evening today. So for the coffee, it's a little bit too late, but it's up to you. You can, of course, drink coffee, but I will prefer drinking tea now. So when you drink tea or coffee, your cells in your body are actually doing something. They changing transcription of genes, right? And what you can measure, eventually, if you have your cells to measure, for instance, blood cells or other cells, you can do transcriptomics, applying various tools like RNA-seq, and you get to know which genes are differentially expressed. All right. From them, you might ask a question, what are the regulators of those genes? So why they're differentially expressed? And to understand that, we have promoters or other regulatory regions of the genes, or even we have additional data maybe about epigenomics, which tells us exactly where the transcription factors are binding. Well, not really exactly, but in a broad window. Or they will tell which areas are open for the chromatin, maybe. And then what we'll see today, using TRANSFAC, for instance, you can find those motifs which are overrepresented in your promoters or in your regulatory regions of your genes. And then from these motifs, you can figure out which transcription factors are involved. And then the topic today is, what is next? The next is to understand what is common between these transcription factors. For instance, is there any cascade of events which eventually can be, we can help to find also using proteomics data, or maybe if you don't have proteomics data, we can actually get it from the knowledge we have collected in the database. And maybe we can build a sort of a cascade of events which happening upstream of transcription factors and leading to activation of these factors altogether. And then maybe there is one or few master regulators, we call them. So such molecules in a cell, maybe receptors or other molecules, which actually change now after I drink the tea. You know, they got activated through some ligands or through other interactions with other cells, maybe. And then they trigger a cascade in the cell, which in fact triggers those expression of the genes. All right, so how we do predictions of binding sites, that we were talking a lot in previous coffee breaks. Just to repeat, so we have TRANSFAC database, which have a lot of data, but a very important part of it's called positional weight matrices. And we have more than 10,000 of them collected over the many years for almost all transcription factors acting in human. And also, well, we cover quite a lot of transcription factors also in mouse and rat and other mammalian genomes. So what are these positional weight matrices? So these are actually such matrices which calculate frequencies of nucleotides in particular positions, which help us actually to predict binding sites in the promoters, what we discussed before. So for that, we use a particular formalism. So we have it in match tool. It's called match, which calculate this Q score. And if this Q is higher than a particular threshold, we claim this is a potential binding site for this transcription factor. The second part of the prediction of enrichment of binding sites in promoters is taking into account their composition. And it's important to understand that very often transcription factors are actually acting in combinations, like in this example. So where 5 transcription factors actually acting together to switch a particular transcription of a gene in very special conditions when all these factors are getting active. And we have tools in our platform, you'll see today, which predict these combinations of binding sites. And at the end, these combinations can be different in different contexts. So in one cell type, the same promoter can accumulate transcription factors over one combination, and they together may activate the gene. The same promoter may retrieve or recruit other transcription factors, maybe even repressors, in another cell types, which lead to the silencing. So, and this change of binding actually requires the fuzzy binding, as we call, so which means this binding is not very sharp, not very strong. It's very flexible. So, and that's why those motifs are also very flexible, that you'll see today. So this is a tool which finds these combinations of binding sites in the set of promoters. So it's using this genetic algorithm, and we talked about before. But now the topic of today is, okay, we found transcription factors, we found the motifs, their combination in the genes which are acting after me drinking tea, or maybe even in more, more stronger conditions in a particular disease state, maybe in cancer cells. They have a lot of genes which are changed in cancer if you compare it to the more normal, not disease state. So we would analyze those promoters, reveal those motifs, and from them we come up with this with a set of transcription factors we predict important for this process. But now the question is there any master regulator which control activity of all these factors, or maybe majority of them? And to do that, why it's important? In case of disease, for instance, you may ask a question whether there's some drugs which may be active on that master regulator, and then through that one master regulator, or maybe few of them, we can control majority of factors, and from that we can control activity of like thousands of genes. It's a hierarchy, it's a pyramid. You target one master regulator and you get effect of thousands of genes. How we do that? So we have a network, a huge network which was collected over many years in a TRANSPATH database. I mentioned it in the very beginning. It's a database where we collect reactions and the molecules which we see in the reactions, and then we actually can build such a network of those reactions to to understand how the signal transduction is going on in the cell. So at the end here at the bottom we see transcription factors. They are blue guys, and then we search for master regulators to this cascade of events. So we consider this network as a kind of a standard graph, and it's a classical graph theoretical algorithm when we actually search for shortest path through that graph, and we actually search for several shortest passes which converge these transcription factors to one or few master regulators. Let me check. I'm getting some messages. Doesn't matter. Okay. So, and through that, we can predict master regulators. So how we do that? We actually compute those shortest passes from my set of transcription factors to all potential molecules in the network. And then we give a weight. We give a particular score, I would say, to each of that molecule. And the score computes actually how many transcription factors from my list can be reached in a particular number of steps. For instance, I say I'm interested in a particular number of steps, which is called radius, how many steps you go upstream. And then you divide it to other transcription factors, which can be also reached with the same radius, but they are not from your set. So we would like to increase this number and decrease this number. So we would like to find such a master regulator which cover majority of my set, but very few of other factors which are not belong to my set. So, and there are some weighting factors here, and basically we calculate the score, and we give the score to each of the master of the molecules in my network, and then we simply rank those molecules and see those ones which are at the top of that score have maximum score, and then these are potential master regulators because they have the biggest influence on majority of our transcription factors. Why these nodes are of different size? Because actually this network is also can be weighted. So it's you can give it weights to these distances between the nodes, kind of virtual distances, which depends on the importance of that protein which we have here. What is importance? Actually, now we can take proteomics data if you have them from the same experiment, which tells us which proteins are actually also change their expression in the disease state. And then we give additional weights to those proteins which are observed in that experiment and give a smaller weight to those proteins which are not found by proteome experiment. Basically, we can, of course, simply remove them from the network, which we are not doing because if they are not found by proteomics, doesn't mean they are not there. So proteomics has a certain sensitivity, and we simply give a bigger weight to those which we found by proteomics and a smaller weight to those which are not found. And when this graph have additional weights, then the algorithm is essentially the same. It just searches not the number of steps, but it's actually calculate the distance. It's similar to a navigation system which you have in your car, and you search for a distance from A to B, and the same algorithm working here. And the third actually concept which we will consider today, and we consider important for analyzing especially data coming from various diseases, and especially cancer, which we have a focus today, is we call it walking pathways. That's a very interesting, strange name, but what we mean is essentially this pathway rewiring. So maybe you heard about this term when there is a standard pathway which may be you can see it in your poster on your screen, or from a database when, for instance, EGF binds to NGF receptor and a particular set of transcription factors and particular set of genes is up or down regulated in normal cell. But in a disease, your pathway may start changing. Actually, things get rewired, especially in cancer. What can happen that another master regulator can take over the job and hijack basically the whole system and start activating a number of genes through a set of regulators. And the special point here is that those kind of bad guys, the pathological master regulators, they are usually characterized by so-called positive feedback loops. So they got entrapped by their own activation. So they activate the genes which are in turn encode components of that particular protein. So for instance, this gene simply encode that receptor. And then the receptor, while being activated, activate the gene, which start increasing expression of that receptor by itself. So the system get in self-replication and self-activation, and that's what happens in cancer, for instance. Then cells start rapidly proliferating. All right, so that's what we see, that the healthy states differentiate from diseases via these feedback loops, via these working pathways, we call them. And that's a characteristic of the pathological state, this number of feedback loops. And that will be our feature to see how to find the real master regulators, the master regulators which are important for your disease. All right. So with that, I will go online. If you have questions right away, just drop them in Q&A. If you have bigger questions, also drop them in Q&A. Maybe I will come to them at the end. We have enough time at the end to discuss your questions. All right, so now we will go online, and so for that, I start from the website of geneXplain, where here we have a button for login. And if you have your account, you can click here and log in. If you don't have your account, you go here to register your account, where you can also, if you don't have yet your license, then you can go and get your license. But once you get your license, you have to go to register here, and with your license key here, you can get your account. So that's important. Before you start working, you have to, get your account. So once you have your account, like I have multiple accounts, but this one for the demonstration. And you sign in. And here you have a set of products which you are licensing to. And it's a little bit, maybe not easy to see, but actually it's very simple. So we have here the database collection of all databases which we have. TRANSFAC, TRANSPATH, and human PSD. We talked about before that TRANSFAC is about sites, TRANSPATH is about pathways, and human PSD is about diseases and drugs. And we have tools like geneXplain platform and Genome Enhancer. We will talk today about these two tools. We have other tools, smaller tools, like MatchSuite, which is for prediction of binding sites. So like the best tool to start with your gene or with a set of your genes and predict, most probable, most reliable binding sites for transcription factors, taking into account as actually a tissue where you're asking, your expression of your genes. But now today we'll go to geneXplain platform, and the geneXplain platform is a platform. Actually, it's a platform of many tools. It's like a toolbox, more or less, which are put. It's not one box. It's these are smaller boxes, like you see on the screen, And our tools are put in those smaller boxes in different combinations. And these smaller boxes are about different types of data. What you're going to analyze, for instance, you analyze RNA-seq data, we are talking today, or you're analyzing epigenomics data, so those peaks, ChIP-seq data, or maybe genomic variants. That's another topic we had also before. Or you would like to actually work together with RNA-seq and proteomics. That's also possible. And that's what we are going to see today. So when you click here on data, you see your projects. And I have a number of projects here, and we'll work in this project, which is for evaluation, where we'll demonstrate how to work with the tool. So the example today, which I want to show, is coming from that paper, which is this set of data, which is not very latest. It's from 2015, but the data are very interesting. It's actually a cell line from osteosarcoma, which was modified. And they put an inducible MYC gene there, so which simulate activation of osteosarcoma, metastasis processes. And there's a paper about that. And actually, what they have here, they have RNA-seq data already pre-processed. So basically, you have here FPKMs. And you can download it directly from here. Actually, in the same paper, they also did proteomics, and it's not in this repository. You can go to the website of that paper and get it from there. So basically, I did it before, and I got this data. So actually, this is transcriptomics data, which they provide in the paper. So that's you see these were six different conditions, samples, and this is a whole set of genes with measured FPKM values. So it's normalized counts, and these are the genes. So that's a typical RNA-seq data, and then we have also here proteomics data, which is another Excel file, which have actually uniprot IDs here and expression of the proteins. It's in fact already a relative expression. It's a log fold change. So it's a logarithm of the fold change, and if it's a positive, it means this protein has increased in the expression in the MYC stimulated cells, and it's decreased in if it's minus. So we have these two tables. And we now go back to our platform. Where is it? Here. Okay, this Zoom things is covering my screen. Anyway, so here we first can create a folder. Let's create a folder. Let's call it DMO2. And we simply upload these files in this folder. Let me go to my computer. And here I go to my folders. Let me see where it is. It's here. Right, so I first will upload the gene expression data, RNA-seq. I select it, automatically recognizes the format, and by the way, you can see various formats, but it usually recognizes the format correctly. So it's a table, and the data are starting from a second row, and in the first row is actually name of the columns. So the first column is the ID column. It's important to select one column which will be your ID. And it's already recognizes that it's an ensemble IDs and it's a Homo sapiens. So you can simply start and upload your table here. So you see now your table here in the major screen. And here you can sort your values by clicking just on the top, on the header. There is also a number of filter possibilities, so that we'll talk about that today. All right, so then we need also to upload the proteomics data. So I'll go back here. I select the proteomics. And as you can see, it's also recognizes all well, and it's now understands that's UniProt. We got our table. All right, so what next? As I promised, next it would be to find binding sites for well, actually, first would be to find differentially expressed genes, and I will go for that. So let me just show you how to find differentially expressed genes. So you basically go to the start page, and here in RNA-seq, you find differentially expressed genes, and you can do it with full RNA-seq data. Actually, it's yeah, you can. It's already preprocessed data, and you can go directly to, for instance, AJAR, where you basically do you take your table, you put it here, you select your experiment, and the experiment is your MYC stimulated cells, and the control is the controls. And then you run it, and it goes now to use AJAR to detect differentially expressed genes. So that's it. So we have log-fold changes, p-values, and adjusted p-values. We can sort by adjusted p-values. You can see there it's quite a good data set. So a lot of the genes which are differentially expressed. So, and which ones we would analyze now. Let's select the most straightforward differentially expressed genes by filtering by FDR and also by log-fold change. So we go to filter, and we put here that log-fold change should be above, above, for instance, 1.5, and FDR should be below 0.5. So that's conditions which we want to have here to get our set of upregulated genes. So we have actually very few, 37 upregulated genes. Well, I would say maybe this condition was too strong. Let me change it to fold change above 1. And then maybe we have more. Yeah, we have 123 genes, which is good. So we can save them with this button as upregulated genes. All right. Actually, we also need to find binding sites for the enrichment of binding sites. We need also a background set. Actually, I have a question here. For RNA-seq, can TPM be used as well for the analysis you are going to show instead of FPKM? Yes, sure. As you can see, we have here various tools. We have AJAR, which works fine with any values. Especially, it's good to work with directly with counts. We actually have also Lima, which is a nice tool, and in Lima you can use different categories with normalized or unnormalized data, and whatever values you have, you can use it to in the platform to detect your differentially expressed genes. Thank you for your question. All right, so now we have this tool, and as I said, we need also another set of genes, which would be our control. And what is actually a control? Let's skip this. We again have these 9,000 genes, and the control is actually the same in the same cell there are a lot of genes which didn't change their expression. So they were in the same condition, but for some reason they didn't change expression. It means their promoters have something which doesn't have something which upregulated genes have. So then let's select these non-change genes as our perfect control. And to do that, we have we have here inside range. So you can put here log fold change and say it should actually be in the middle, so around 0, from minus 0.1, for instance, to 0.1. Let me see how many of them we have. Yeah, we have 1,700. It's a pretty good control. This is quite a good set of genes which didn't change the expression at all. So their fold change is very little. And as you can see, the p-value is also, it's around 1 or 0.5, so which means it's not differentially expressed. So now we have these two sets of genes, up and non-change genes. And to detect, to find motifs, we go back to start page and go next, where we want to find, basically analyze regulatory regions. And we want to identify enriched TFBS in promoters. So we click TRANSFAC. And now that's, something which you haven't seen yet, maybe. This is a so-called pipeline or workflow. We call it a workflow. So, and the workflow is a set of tools where you can see it here below. The tools are here blue, and they're connected by the input and output. And this is a long workflow. So what we first do, we put here our set of genes upregulated. And by the way, if you don't have any control, so for instance, you just got your set of genes and you don't have anything to select non-change genes, then you can use, of course, universal control, which is promoters of housekeeping genes, which, as usually people use it, but although it has certain problems, because housekeeping genes are also changing, in fact, in many conditions. But okay, if you don't have anything, then housekeeping gene is the best approximation. But we have these non-change genes. We can put them as a control, and we call it yes set and no set. So, which in other words, like foreground and background. Now it's a human, and the annotation is also taken from human. The profile is a set of positional weight matrices which we want to work with. And in fact, if you click here, you see all possible sets of matrices which we want to which you can select. And this one, which is here by default, is actually the whole set of matrices from a vertebrate. It's around 7,000 positional weight matrices, so the complete vertebrate set of matrices. So we want to apply them all. And then there are parameters like what should be your distance of analysis of your promoter. And as I said, here we don't have data about which region of promoter is actually acting. So we don't have cheap-seq data. If we would have it, we would use it for defining which region we want to analyze. But since we don't have cheap-seq data, we define here from 100 downstream to minus 1,000, and we run our workflow. So, and as you can see, sorry. As you can see, this workflow shows how it is working. So you can monitor how it is progressing, and at the end, it gives you the data. So there are a number of steps to find binding sites, to find combinations of binding sites. You also can look at the previous coffee breaks where I was actually in detail describing how to do that. Today, I want to show you some results already of this site prediction, because the major topic of today, we want to talk about pathways, right? All right, so as you can see here, I have actually the run the Lima search instead of Azure, so it's a little bit different. And I analyzed this enriched upstream upregulated genes. Let me see where I have it. Yes, here. And this workflow, which is running now, it returns a set of predicted motifs, which you can see like that. So you will see, and what I did run, such a set of motifs, set of positional weight matrices, which are, here we found 352, which are sorted by p-value. And well, we can maybe sort it by the yes to no ratio. That's maybe more interesting to discuss. What is yes to no ratio? So, okay, so it's already the data upcoming from that workflow. When workflow is finishing, it automatically opens the result tabs like a report. Okay, let me maybe close this and talk about what I want to show you today. That's a final report which this workflow generates. By the way, such a report with all the motifs which is found in a nice HTML format. You can also download it as a PDF for your future consideration. So you have such a table where this yes to no ratio shows to which extent this particular motif is enriched in the promoters of upregulated genes. So if it's 6, means it's 6 times more frequent in the promoters of upregulated genes compared to the promoters of non-changed genes. And the p-value of that difference, of that difference in the frequency is significant, in -5, right? Although these motifs they're quite rare by themselves. So this gives you the frequency of each motif in the promoters, and it's 0.3, which means only 3% of your promoters contains those motifs. But in the null set, only 0.5% contains promoters contains these motifs. So they are rare, but they're enriched. There are other motifs which are more frequent. For instance, this motif, which 14% of genes or even higher. So if you look at this motif, for instance, they even many motifs in one promoter, like 4 in average, 4 motifs in the promoter or more, but in the null set there are less number of them. And then the p-value, of course, in this case is more significant. All right, so we have these motifs. And then when you look at them and these motifs, you can, in fact, look at them visually on the tracks. So first, let me show you how to visualize those motifs very easily. So let me, for instance, sort it by the yes to no ratio, select some of the motifs I'm interested to visualize, and then click here this visualization button, which gives you a sort of an overview of those motifs in each promoter. So, for instance, in the first promoter of that gene, we see these motifs of those top ones which we just selected, and there's 11 motifs, and you have here the table of how many motifs of each positional weight matrix was found in that particular promoter. And then you see here another set of motifs found in this promoter. And by clicking on each of the motifs, you can also see them here, what was the consensus and what was the cutoffs and so forth. All right, so once we have these motifs, that's fine. But what is the next? Next, we want to know which transcription factors are actually enriched here. So motifs for which transcription factors. And this workflow, it actually gives you this set. So it converts these motifs on the fly to the set of transcription factors. And now you see these 175 transcription factors, transcription factor genes, which are whose motifs are enriched in promoters of our upregulated genes. And as you can see, some of the transcription factors, like this one, is connected to several motifs. As you know, in TRANSFAC, we actually collect a lot of motifs from various literature, from various conditions. And one factor may have quite a number of motifs, number of positional weight matrices in TRANSFAC. But each of the positional weight matrix for one factor actually is a little bit different. It shows a certain focus on that factor in maybe in one or another particular condition where this factor is binding to the target genes. So other factors have just one motif. So with this, you see this list of factors. And what is next is actually, well, next we can annotate those factors. Well, they're already annotated. And we can also add to them expression, if you like. Let me open that another data. So adding expression or adding any other annotation is very easy. So for that, we have this annotate table functionality, so you can find it by yourself. So where you put your table and you put another table which you can use to annotate your original table. So at the end, we have this with this set of transcription factors already filtered by the expression. So again, so as you see, we have factors which are associated with your motifs, but we don't know whether those transcription factors are in fact expressed at all in that cell line. So, but in fact, we have RNA-seq data which shows us almost all expression of all genes. So that's why what we can do, what we are doing here, we annotate our list of factors by the data, by the expression data from the same experiment. And now we see those factors which are not only associated with enriched motif, but they also differentially expressed by themselves, like MYC, for instance. Obviously, because MYC was activated through, artificially activated, and we see this expression of that gene very high, and we see a lot of MYC motif in the promoters of upregulated genes. But we see also other motifs, other factors, which are differentially expressed, and their motifs also enriched in the promoters. So what we did actually by doing filtering of that table, I filtered those factors which have a very high level of either upregulated or downregulated. Downregulated is also interesting because this factor is downregulated, so its presence is changed. And the motifs are enriched, and sometimes this factor may play a role of a repressor. And if it's actually amount of this factor, activity of that factor is decreased, then those genes which have motif for these factors will be activated in case if this factor is actually playing a repression role. So at the end, we got this 37 transcription factors, which are very good factors. So their motifs are enriched in upregulated genes, and they are differentially expressed by themselves. All right. And now, why do I need that? I need to search upstream from that. Now we come to this upstream analysis to finding master regulators. And for that, we have to return home. And here you see workflows to detect, to analyze networks, right? And to find master regulators. And here we have this TRANSPATH. We have also other databases like GeneWays. And with TRANSPATH, with single gene set analysis, we can analyze now this set of transcription factors and find master regulators which are controlling these 37 transcription factors. So we will take this gene, we put gene set of factors, we put it here. We select, very important, the species, because actually different species have different network. So human, mouse, rat, these signal transduction networks, generally very similar, but there are a few differences. So that's why when you select human, you are working with a network of human, and then you run this workflow. So it takes a bit of time. That's why I will show you the result of that workflow search. Yes, I think I ran several of them, and I will show you this one. No, let me show first this one. Actually, what you see here is a list of keynotes, we call them, which are sorted by the score, which I mentioned in my presentation. So this score is, the higher the score, the more chances that this molecule, we don't call all the 5,000 key molecules which are found here as master regulators, because actually this just shows the complete set of all molecules which can be reached from where your transcription factor list can be reached. So you have to put the cutoff on the score. But at the top, we see, for instance, the proteasome as a key molecule, and we can, in fact, we can visualize it. Let me visualize it by clicking here, just to show you how does it look like. So actually, this is a list of transcription factors in the table, which you see here. So these are transcription factors from my list, from these 37 factors. And in fact, not all of them can be reached from that particular key molecule. So only 19 from 37 can be reached. But it's a very big number, so it's more than half. And actually, that's the maximum number which can be reached in this network. So not all transcription factors which can be predicted on the level of motifs can be, there are data, there's knowledge about their connectivity. And when we predict the majority of them, and we predict a master regulator which can control them, then we are already happy, because it's like it's a paradigm like a big elephant in a room. So we see an elephant, basically in this case, this proteasome, which may change activity of this number of factors. So here you see the blue guys are our transcription factors, and the green guys are the intermediate molecules. So this signaling goes from proteasome to another kinase, and then to another kinase, and at the end it phosphorylate or activate through other means the transcription factor, which then start binding to your genes. So we have this cascade, and we predict this proteasome as a master regulator, although it not covers all 37 factors, but it covers majority of them. And that's one big elephant in the room, which triggers a lot of factors, and it gives us evidence that that's the guy, that's the most important master regulator. But in fact, we have to put a certain cutoff on that on this score, and not just consider the first one or the second one. We should consider more of them, because it's all just predictions, and depending on the score parameters, we may have more or less of those factors together. That's why at the end we are talking about set of potential master regulators. We can visualize them all together. Like in this case, well, this is just one. But if we select first 50, like in this case, so you get such a network where you have at the end your transcription factors, and then at the top the master regulators, which you selected the 50 top, and we know that many of them, or maybe all of them, are important to trigger the activity of that factors, and then the end activity of target genes. Still, we don't know exactly which one is most important. So that's another task to figure out. And to figure out which ones is more important, maybe we should remember what I talked to at the very beginning about these positive feedback loops. So some of those active master regulators are actually produced by the genes which are activated by these factors. And how to find these master regulators with the feedback loops? For that, we have a number of workflows. So actually, here we have here at the end, upstream analysis with feedback loops using TRANSFAC and TRANSPATH. This one workflow actually goes all the way from the list of genes and it searches for transcription factor binding sites, detects factors, filters them, runs upstream analysis, and actually searches for master regulators. And that's one workflow which is quite big. Actually, we have a bigger workflow, which I will not talk today about that, just to mention. We have a tool called Genome Enhancer, which does what I said, even with together with many other data sets. So it can combine transcriptomics, for instance, with proteomics. It's called Genome Enhancer. And then in the Genome Enhancer, you get results with the feedback loops, with proteomics, with everything. And let me just show to you the result of that Genome Enhancer work. Basically, here this is a master regulator. Let me see here. So this is a network with master regulators which have a feedback loops. So you have here, you see these dotted lines, which goes from the genes in the bottom. And in this network, we have not only the transcription factors, but we also have these target genes, those genes which encode for the master regulators. And this pipeline of Genome Enhancer can also take transcriptomics and the proteomics data, which I showed at the beginning. So we uploaded it, but not yet used it. But Genome Enhancer can use it. And then it actually takes into account when searching for the shortest passes through this network. And then it shows here on this shades around the nodes. So the left part of the shade is coming from transcriptomics. So if it's red, it means this gene is upregulated. And the right part coming from proteomics, it tells that this protein is upregulated in proteomics. So you see this one in transcriptomics, this one in proteomics. This complex, actually in transcriptomics showed downregulation, in proteomics upregulation. That happens. But often, especially among these master regulators, you see both transcriptomics and proteomics, they go in concordance. So they showed these master regulators, they upregulated on transcriptomics level and on proteomics level. So they are the best molecules to choose for as a hypothesis about which master regulator is actually regulating your gene. All right, so actually I wanted to do a little detour to Cytoscape, but let me first answer the question. Here we have a question. This network or pathway is generated through in literature or specific for the experiment? A very good question. Actually, both. Of course, each of that reaction in this network comes from the literature, and you see here what kind of literature. So you click here, and you see what was the reaction, what was the database reference, what is a PubMed reference. So from which paper this particular reaction was annotated. And I must say, it's a huge work. Our curators went through a lot of literature, and at the moment, when we count how many reactions we have in our database, it's more than 1 million, which was, well, partially by hand, partially through, some text mining tools. With the help of text mining, we collected this information. Each reaction comes from the literature, but the connection of reactions, like this activate this, but this activate this, maybe never published. So this connection is found by the algorithm, and it's found based on the data which we are analyzing. So and the answer is both. It's both from the data and from the literature. It's kind of a combination of knowledge, prior knowledge, and the data, experimental data which you have in your hands. Another question. Okay, that was the question. All right. Okay, let me show very quickly how we can do things in the Cytoscape. So for that, what we do, we actually can export this network to your computer. So you have here the button, export, and there are a number of different formats which you can use to export. Of course, you can export just as a picture to put it in your presentation. And you can export in various formats, and as BML, systems biology, makeup language, GraphML, combine, and for Cytoscape there is a special format called CX, and we export it as a file. And now we go to Cytoscape, and actually that's already pre-processed data, but let me show you how to put this file into Cytoscape. Basically, you go into import and the file, and you select this file. It asks you where to put your file, your network. You type OK, and bingo, you have your network here with a preserved actually layout here. Although it's a little bit crowded, so basically what we have here, we have nodes for the both for the genes, for the proteins, as well as for the reactions. So we have reactions, and the reactions names are quite long. So we can get rid of this long by various things. For instance, what we do here, we want to remove labels. So actually we click here on bypass. First, we actually have to select the nodes which are for reaction. So to do that, we go to select, and here it's in the kernel, in this table of all nodes here, we have this kernel type, and we type here reaction. Sorry, reacting. And with that, we actually selected all reactions. And now we go to style, we go to bypass and click here, OK. So you see there's no names for the reactions. They are just names for the proteins. Here we have actually genes, and we still have reactions. Okay, so we can move our genes a little bit. So we can move here. So one thing which is important, which is very nice in Cytoscape, there is a number of tools which can be applied here to analyze your network. For instance, we're interested to analyze this network and ask a question about different parameters of that network. So you can treat it as actually this network is in fact a directed graph. So it has directions, which is important. It's not just protein-protein interaction, it's reactions from to. So it's directed graph. And it calculates it also a certain thing. I will talk about that a little bit. But it's also give you, information about connectivity. And what is nice here, what it's already did, because I did it before, it's already remembered what I did before, it actually give a different weights to our network proteins. and give a different size according to the betweenness centrality. Why I'm talking about that? So you remember we end up with a set of master regulators, which were actually a set of 50 master regulators. Now it's still quite a lot. So we want to, there is another question. Maybe there is a certain hierarchy between them. Some of them are maybe more important, some of them are less important. And one of the parameter of importance is called betweenness centrality. So in the graph, actually it tells the higher the betweenness centrality is of those nodes, which are most important for connection between other nodes. So if you go from one master regulator to the transcription factors, you will go through some nodes. We can call it hubs, but this is not really a hub in a formal definition. It's a special role which is called betweenness centrality, and it's very important to such nodes which have such important position in the network. They are now get a higher size, and by doing that, you see among those found master regulators those ones which are actually indeed very important. And among them, of course, we see MIC. Of course, we see MIC because it was the one which was activated. But actually, we see also EGR1 as an important one, and well, maybe less important, but still quite important. We see these master regulators like FKP-BP4. So this gives us a chance to select better, few hypotheses, and then go to my experiment and do my verification of those master regulators. All right, so I think with that, I want to finish today my presentation, my demonstration. But we have still, some time if you have time to ask questions, and I'm ready to answer. So don't be shy. By the way, if you are not online, but you're watching this video in our YouTube, it's a time to give us, like. The more like you give, the better we are in the YouTube, promotion. All right, so I hope it was interesting for you to learn about this platform. You can register for the platform. There are various ways of getting licenses or just to get free registration. So you just go to our website to see what is possible, and then you have plenty of possibilities to analyze your data. All right. Okay, so if there's no questions in between, then thank you very much. But you have there's one question. On a moment. Okay, thank you. Does TRANSFAC have transcriptomic data like ChIP-seq data? Transcriptomic data like ChIP-seq data. Yeah, so the TRANSFAC, yes, TRANSFAC has ChIP-seq data. So we are one of the biggest collection of the ChIP-seq data. So we collect peaks from various sources, from ENCODE, from other databases, from literature directly, and in TRANSFAC, if you go to TRANSFAC database, I'll show today the platform, but as you can remember here, we have this TRANSFAC and the TRANSPATH, the whole set of databases with the search engine. And here you search for your transcription factor or gene of your interest, and then you can go to this gene and read a lot of information about this gene. And also you will see ChIP-seq data about this gene, about this transcription factor. So for instance, BRCA1 is a transcription factor. Well, although it's a not very classical one. And in this case, we have, we have information about binding sites for this factor. And do we have a ChIP-seq data for this factor? Maybe not, because it's not classical one. No, in predict enhancers, we have enhancers. Yeah, here, in vivo fragments. Right, so we have ChIP-seq data for BRCA gene. Actually, it's these are ChIP-seq, sorry, it's a ChIP-seq data from, so these are peaks of other transcription factors which are found around the BRCA gene. So you see it's, peaks around the BRCA gene in the promoter of BRCA gene. But you can also search by the factor. So that you can search by a gene or by a factor. So we have indeed ChIP-seq data in our database. And this ChIP-seq data can be used in inside our tools. You can, for instance, one of our tool, which is called Match Suite, it has a possibility to visualize the ChIP-seq data on the promoters of your predictions, and also you can use it in the platform to filter your results. All right. Okay, if you don't have more questions, then we can finish for today, and send us questions. If you send us questions via our forums or just directly by email, we'll try to answer them in the next coffee break. So we'll send you the answer right away by email. Okay, thanks a lot, and see you next time.